Welcome to the second in the series of 3 ELIXIR webinars focused on collaboration with Romania. So this second session that you're joining this morning is focused on collaboration around human genomics and translational data. And we're doing this event jointly and in conjunction with the Romanian Society for Bioinformatics. So my name is Andrew Smith. I'm Head of External Relations at ELIXIR, and I'll shortly give you a very brief introduction to what the program is, so some of the themes and speakers who will present to you, and then I'll also give you a very quick introduction to what ELIXIR is itself, before we then open up into the presentations about the various opportunities for engagement in ELIXIR around human genomics and translational data. So the program itself, so we have a series of talks from representatives of the ELIXIR Hub and also of ELIXIR nodes, as well as Romanian experts as well. So it's a 2-hour webinar, and we really like to make this interactive. So it's a chance for you to ask questions, and I'll explain in a moment the different ways that you can do that. But if we look at the program, in a moment, Gary Saunders, who is based in the ELIXIR Hub and coordinates ELIXIR's work around human data, he will give you a presentation about the activities and vision for how ELIXIR wants to coordinate these activities across Europe. He will also mention initiatives like the 1 million genomes initiative, and also things like the federated EGA. So these are activities that Romania can be aware of, can participate in right now. And so that would be a longer talk because it's quite a major set of activities that he's describing. I think we'll take a break in the middle of that presentation. We'll probably pause halfway through to give you a chance to ask some questions before Gary continues his talk to talk about the federated European Genome Phenome Archive. Then Kathy Lauer, who also works in the ELIXIR Hub as ELIXIR's industry officer. Well, as well as that, she's also coordinating ELIXIR's activities around coronavirus. And so she will give you a presentation about what ELIXIR is doing to help in these areas. And also that will give you a good idea about opportunities for engagement as well. We'll then take a short break before Hedi Petersen, who is the Deputy Head of Node from ELIXIR Estonia. She will give you a bit of a presentation about what ELIXIR Estonia looks like, and also the human data related activities and resources that they have in Estonia. Before Cristian Zimbru from Timisoara will give a presentation about, he will offer his perspectives about the Romanian Human Genetics Laboratory and some of the local activities that are taking place there. Before we open up to have some of the specific human data related talks, for those of you who didn't join the first presentation and have just joined this second webinar, I thought it was worth just recapping some of the basic bits of information about what ELIXIR is. So ELIXIR is an intergovernmental organization, and its aim is to coordinate, integrate, and sustain life science resources. So these resources include things like databases, so repositories where, as a scientist, you can deposit your data. It includes resources where you can get data from, so value-added, curated knowledge base resources where you can access data for free of charge. They include software tools, analysis tools, and workflows that you can use. It includes training materials, so sometimes face-to-face courses, a registry that allows you to find resources, as well as online training. We have a lot of interoperability resources and standards, so things to make your data fair, which are also available. And there are lots of national computing facilities that you can use to analyze data, as well as local data management support, so experts in countries that can help researchers and consortia with their data management needs. So they are the types of services that ELIXIR offers. And what we want to do by connecting across countries and across institutes is to try and coordinate this. So you go from having hundreds of fragmented resources to one single coordinated infrastructure. Because we are an intergovernmental organization, and these countries that you see on the left are all formal members of ELIXIR. So they make a contribution to the ELIXIR Hub, which gives us a budget to spend on projects to connect ELIXIR nodes together. Romania isn't yet a member of ELIXIR, but in the long term, that could be an option, and that's something that we would like to consider. But throughout the rest of this talk, you'll also hear about opportunities that you can benefit from even without being a member as well. Because once a country joins, it creates an ELIXIR node, and it's those nodes themselves that run the services that are part of ELIXIR. And over the long term, we would really like institutions in Romania, after it becomes a member, to create an ELIXIR node and fully take part in those activities. And then before we move over to Gary's talk, it's worth just explaining the focus of ELIXIR in the context of the structure. So we have 22 members and an observer. Each of those countries create an ELIXIR node, which is their national bioinformatics infrastructure and how they support local users. Well, at the European level, we then try to connect those experts together in particular areas. So we have 5 platforms which cover tools, compute, training, interoperability, and data. And they bring together ELIXIR experts in those areas. And we also have communities, and these bring together ELIXIR experts in a particular domain. So they're domain specific, including things like rare diseases, federated human data. And you'll hear about some of those today through the talks. And they also include opportunities for researchers from outside ELIXIR to take part in those as well. So that's what I wanted to stress to you today, the focus on those human data communities. That's where I'll finish my talk. I probably have time for maybe one question before we move into Gary's presentation. So if anyone did want to raise a hand, this is a chance to ask a general question about ELIXIR, if anyone has any. No, it doesn't seem to be the case. In that case, I'm going to ask Gary, our human data coordinator, if you can show your slides and begin your presentation now, please, Gary.
Okay, thanks, Andy. I hope you can see the screen and hear me okay.
I think Gary's on mute. Can you unmute Gary, please?
Can you hear me now? Excellent, thanks, Andy. Okay, so my name's Gary Saunders and I'm based at the ELIXIR Hub. And in this presentation, I'll take us through the activities that we have on human genomics and translational data. But as Andy explained, it'd be really great if we could have questions at any time or discussion at any time. So please do feel free to interrupt me. Otherwise, we'll have 40 minutes of me speaking non-stop. So I'll ask my colleagues at the Hub to keep an eye on the chat, and if any questions do come up, we can stop and have a deep dive into any topic. So at a high level, ELIXIR Genomics and Translational Data aims to build a virtual cohort of human samples using federated, secure, cross-border technologies. So I'll come back to those themes time and time again to explain really what we mean. But the potential here is that we could have a virtual cohort of around 60 million human samples by 2025 at the current rate of genome and exome sequencing. And indeed, this rate of genome and exome sequencing is even predicted in many places to increase. So the 60 million cohort might actually be an underestimate. There are going to be many challenges to try and build that virtual cohort, not only because of the wealth and the amount and the volume of data that we're talking about, but there are many challenges that must be overcome to try and create a cross-border infrastructure for accessing sensitive human data. The main reason in my opinion, and in the opinion of ELIXIR, is that we're living in a time where the generation of human genomics data is shifting from the research space to the healthcare space. And as the healthcare space across Europe and indeed the globe generates these data, healthcare data is a national competence and is held much more securely than classical research data. So we know that the data that's generated will be held within national jurisdictions and very likely in many cases won't be able to leave. So we have to find a way for researchers, clinicians, other healthcare systems to try and access these data from wherever they are across international borders. And that's where this phrase at the bottom right here, federated, secure, cross-border access to personalized medicine data, the vision of ELIXIR Genomics and Translational Data, that's really where it sits. So in this presentation, I've tried to phrase it in 6 distinct parts. So the first is to explain why I think sensitive human data needs this federation and needs this federated infrastructure. And then we'll have a look under the hood at ELIXIR. What are the structures of ELIXIR human genomics and translational data? Then we'll discuss some key international collaborations that we have from ELIXIR. The examples I'll focus on are beyond 1 million genomes and our collaboration with the Global Alliance for Genomics and Health. Then we'll delve into a bit more technical discussion to look at what technical solutions ELIXIR is offering for data federation. And then at the very end, leading nicely into the next presentation from Kathy, I'll discuss the COVID-19 use case for sensitive human data. And then we'll summarize the talk. And ideally, we can have some time at the end for discussion. So these numbers really show what I was trying to describe earlier, that we are living in a time where the generation of human genomic and exomic data are now moving into something that's funded primarily by the healthcare system. So if we look at the percentage of human genomes and exomes that were funded by the healthcare system in 2012, it was as little as 1%. In 2017, this moved to 20%, but the prediction is by 2022 that 80% of the human genomes and exomes that are sequenced will be funded by the healthcare systems across Europe. And we can see the manifestation of these numbers here. So this is a figure that we published last year, and it looks at the genomics-based national initiative projects across the ELIXIR members. So these are national initiative healthcare funding programs in the ELIXIR members, and we have almost 20. So this really shows that we know the data is being generated by the healthcare systems in these countries. But this brings with it a number of challenges. At the top, again, I've repeated the number of the potential of the virtual cohort of 60 million by 2025. But really, if we want to try and access these data, some of the key challenges that are in front of us I've listed here. So the data will be geographically distributed. Clinical data are often right now not interoperable for many reasons. Language would be one, different ontologies to be used is another one, so we have to try and overcome that, and I'll come back to that later. And then the bottom 4 points, I think mostly talk about the technical expertise. So we know that healthcare is not used to this amount of data. So we're talking about terabyte to exabyte volumes of data here. And the technical solutions and the technical know-how of how to handle data at this level, it sits right now within the research community. So we have to try and find a way to build the capacity within the healthcare systems across Europe to explain this technical know-how, to pass on the technical solutions that we have within the research space into the healthcare space. And for this to happen, we have to look at the attitudes and actions towards open data. Now, I'll come back to the Beyond 1 Million Genomes project later, but really that's one of the key projects that we drive from ELIXIR, to try and look at the attitudes and actions towards open data. And by open data, I don't mean that we give everyone access to healthcare data across Europe is that we use the proper authorization and authentication infrastructures so that those who should have access to the sensitive data can indeed access these data. And then at the end, absolutely, we'll need a strong governance model so that we have trust across this federated research infrastructure and across this federated infrastructure to access sensitive human data in order for it to function pan-European level. So I've put here basically 3 figures that I think underpin a lot of the work that we do at ELIXIR. And that is to try and build this federated infrastructure. We rely on the use of interoperable standards. So we don't say that everyone must use the same technological solution, but what we do try and stress is that if we can promote the same standards to be in use, then data are, by proxy, interoperable. We know how data are generated, we know how data are stored, and we know how data can be accessed across different European countries. One of our key phrases from ELIXIR Human Genomics Translational Data is to build a network of trust. So this network of trust underpins the technical infrastructure that we have from ELIXIR Human Genomics Translational Data. Without this network of trust, it's very difficult for us to build the relationships and build the key know-how and share that know-how in a capacity building manner across Europe. And then at the end, we also have reference archives. This refers to something called the European Genome Phenome Archive that I'll come back to later. So now, looking a bit more under the hood of ELIXIR Human Genomics and Translational Data. Our mission is to try and coordinate this sustainable infrastructure for human genomics and translational data at a pan-European level. And importantly, try and expedite the transition of these data into medicine. And the vision, therefore, is to facilitate the discovery, access, sharing, and analysis of genomics data. And these are all factors that I'll come back to later in this presentation. So Andy presented a slide very similar to this earlier. And indeed, for those that joined the first session, may have seen this structure before. So we have the 5 technical ELIXIR platforms in the center here. And we have the array of ELIXIR domain-specific communities around the edges. And by these grey lines here and grey dots, we show a schematic vision of how the platforms can be driven by the use cases from the communities. But the 3 communities I want to draw your attention to here are at the bottom. So we have federated human data, human copy number variation, and rare diseases. And if we look very closely, we can see that actually they're joined by these 3 distinct lines. These 3 communities are brought together by these 3 lines. That's because at ELIXIR we have an additional umbrella structure here called the human data communities. So the human data communities looks to work across these 3 communities to build a coordinated, federated infrastructure for sensitive human data access. But it isn't only those 3 communities that I work with and coordinate and contribute to that federated infrastructure. So this is a bit more detail to the figure that we showed previously, where in the center here we have the human data communities, and we have some flagship projects. The 2 that I've listed here I've put because I'll give more technical information about them later. That's the European Genome Phenome Archive and the Beacon. But it is absolutely vital that we work directly, not only with the 5 technical platforms, but with a number of projects that are coordinated by the nodes. So the nodes roadmaps, the nodes funding, we try and coordinate that into an infrastructure, but also a number of EU and indeed wider grants. So I've put some examples here, the now finished Accelerate and COBEL projects, but at the bottom we've got EJPRD, the European Joint Programme on Rare Diseases, FAIR Plus, EOSC Life, Seneca. Many of you may be involved in these projects or at least be aware of them. The objective of the ELIXIR Human Genomics and Translational Data is to try and coordinate across this plethora of projects so that we can coordinate where we can to find the technical solutions to build this federated infrastructure. On the right-hand side here, I've also put some key partners. So BBMRI ERIC as another European research infrastructure, a very important partner of ELIXIR. IMI is the current funding initiative linked with industry. And then at the bottom, we have the Global Alliance for Genomics and Health. That's a key partner that I'll come back to later. So the objective here is that we know internally at ELIXIR that we have a number of different structures where our nodes contribute and our nodes try and build projects for and try and build visions for, but this tries to encapsulate the vision and the funding and the development in a whole sphere of other places. So if we're looking at opportunities for involvement, then yes, it's great to have as many nodes as possible involved in our communities, federated human data, rare diseases, human copy number variation. Within human genomics and translational data, we also have two key focus groups. And these are relatively new focus groups that started only in the last year or so. We have one on cancer data, and we have one on health data. And the objective of these focus groups is to try and define what the ELIXIR stance is and what the ELIXIR relationship is in dealing with data of these types. So I'll just again stress what Andy mentioned at the start. Primarily, these structures are for ELIXIR members, but within the communities and within the focus groups, we have the opportunity to interact with researchers and clinicians and infrastructure builders across Europe that are not currently members of ELIXIR. There are slightly different rules there, but if anyone is interested in any of these topics and would like to know more about it or would like to know more than what I deliver here, then please do reach out or we can discuss it at any time. So this is a similar figure to what I showed earlier. Again, we're going back to these national initiatives across the ELIXIR members. So these are primarily private funding, or there's a mixture of public funded and public-private funded, but importantly, healthcare-driven national initiatives that are generating genomics and exomic data across Europe. So what we know, again, is that these data are generated in the healthcare system. The healthcare system holds data as a national competence, and it's very likely, in many circumstances, not to leave national jurisdictions, which provides a huge challenge for a federated infrastructure. But I'm pleased to say that in 2018, there was an EU declaration signed by a number of countries. I think we're up to about 26 now. The list here is on the right-hand side. That's the current list of signatories. And this list of European countries signed a declaration that they would like to share at least 1 million human genomes across Europe and across the European Union by 2022. So even though we know that there's a whole array of challenges that have to be overcome, it's excellent to see this political will of many European countries to try and overcome that. So the scientific explanation that I gave earlier about building cohorts of 60 million, that's one driver of this, but there are others. And where I've listed the countries here, what I've put in orange are the ELIXIR members. So we can see that of the signatories, a huge number of them are also members of the ELIXIR research infrastructure. And what we did with many of our European nodes, and also BBMR ERIC as another research infrastructure, along with GA4GH as a standard setting agency, we'll start to write a paper, which was ultimately published in Nature Reviews Genetics last year, to say, if we have this declaration, this political will to access 1 million genomes by 2022, how can we leverage the European infrastructures to aid this? So this paper that you can reach at this link at the bottom here, completely open access. This paper outlines what ELIXIR, BBMRI, ERIC, and GA4GH can contribute in order to for us to access 1 million genomes by 2022. And at the orange, I think I've put one of the most important statements from the paper is that ultimately it relies on a suite of interoperable standards. And I'll come back to more technical information about what we mean about that later. So this is one table from the figure, and I think it's something that we use at ELIXIR quite a lot. So we use it for the human genomics translational data vision. We use it in coordination with our nodes. We use it in coordination with external bodies. And it outlines the 6 key necessary minimal infrastructure components that we need to be in place to access 1 million genomes transnationally by 2022. So it describes the standards, it describes the programmatic interfaces, the secure federated clouds, the tools and services that we need, importantly, the legal and regulatory frameworks, the training materials that we need, and ultimately a capacity building program. So this is not something that I'll delve into a lot right now, but as in future years or months even, as perhaps you become more involved with the ELIXIR human genomics and translational data vision and what we try and drive towards, this table here is a huge underpinning of what we try and focus on. So this is the vision, the federation of data. And this is a figure that I quite like because it explains it in a relatively high level way, where if we look at the map of the globe, then each of these blue dots is a sensitive data archive. So we mandate that, or that we would state that there's a sensitive data archive, many of them across different countries. There may be one national one, there may be more than one national one. But importantly, these sensitive data archives are connected by these lines. So if you can see these lines that connect the sensitive data archive, these should say that the data is accessible from pretty much anywhere else in the globe. So though we know we have these many different sensitive data archives, the data can be accessed and analyzed from many different places. So this is the vision of the Federation of Sensitive Human Data. And at the top, I've given 2 of the key drivers that I see from ELIXIR. The first is bureaucratic, legal, GDPR. That is because we are moving into this space where data is generated in the healthcare system, and it's national competence. It's completely understandable that these data are held more securely. The second driver is that of the scientific case. So an example would be rare diseases. So in our rare diseases community, we know of examples where patients might only have one other patient with similar phenotypes and similar clinical manifestations in a country that is many, many miles away. In order to link those patients together, link the genomics data, the phenotypic data, the serological data, all types of proteomic data, all the different types of data, that's assigned to try and find an underlying reason for that disease. That is a scientific driver for the federation of data. That's one that's growing and one that's very, very common to hear in projects such as the European Joint Programme for Rare Diseases. So in the next part of this presentation, I'll look at the international collaborations that we are leading and building from ELIXIR to try and build this framework of federated sensitive data archives. The first one will be beyond 1 million genomes, and then, as Andy said at the start, I might stop after that description, and then we'll go into the more technical aspects through ELIXIR GA4G8 strategic partnership, and we'll look at the beacon and we'll look at the European Genome Phenome Archive. So this is the first slide to describe the coordination and support action beyond 1 million genomes. So this is a EU funded, Horizon 2020 funded CSA for €4 million across 3 years. There are 28 partners, it's a large project, and importantly, there are 25 European countries involved. So this B1MG project is the first funded project from the EU to implement the declaration to share 1 million genomes by 2022. So what we say, and what is an important distinction here, is that this is not the member state led 1+ million genomes initiative, the signed declaration, but we link very tightly to that 1+ million genomes initiative to take the recommendations that come from this project. Excuse me, I have a bit of a cold at the moment. But we take the recommendations from this project, align them with the recommendations from 1+ million genomes, so that we can use this coordination and support action as one of the key tools to implement the declaration to share 1 million genomes by 2022. So as I said, there's a whole number of partners in this project. It's a large and complex project. So you can see some of the key research infrastructures, ELIXIR, BBMRI, ERIC, IATRIS, and ECRN. And then a whole suite of other partners that we have across Europe in the consortia. And there are key challenges with a project of this size and a project of this complexity. Again, I want to stress that this is the first EC project to support the 1+ million genomes initiative, but it is not the 1+ million genomes. So there are two distinct bodies here. We have the B1MG, which is the project that we coordinate and lead. And then you have the member state driven 1+ million genomes initiative. And although we have stakeholder groups that allow these two entities to talk, and we join some of their meetings and they join some of our meetings, it is not the 1+ million genomes led initiative, but we are tightly linked. The B1MG is governed by the bodies and supervised by the European Commission, and it will support the 1+ million genomes initiative by implementing the B1MG description of action. So that's what the B1MG project will deliver. It's a description of action, and I'll come back to that in a couple of slides' time, exactly what we mean to that. But we do work incredibly hard to maximize alignment with 1+ million genomes during the project. So we know that some of the ELIXIR nodes, their representatives within the 1+ million genomes initiative, are also key work package leads within B1MG. So absolutely, this helps coordination at the pan-European level, but also at the national level for these nodes, where they can have the same people represent on the 1+ million genomes and the B1MG project. So at a high level, what will this B1MG project do over the next 3 years? And I think there are 3 key pillars to this. First, the overall aim is to drive the development of federated and secure cross-border access to personalized medicine data. And the first objective is to define the requirements. So at a pan-European level, what requirements are there to have this federated infrastructure? And then once we have the requirements, which are in progress right now, the generation of them is in progress right now, we will then look at the technical specifications and implementations that allow us to meet those requirements. Now, there's no funding for technical work within the B1MG, but what we can do is leverage existing solutions that meet the requirements. So we can find recommendations and implementation guidelines from the B1MG project to take work from other projects across Europe to meet the requirements for this federated infrastructure. And importantly, what we have to try and do is drive the adoption of these solutions and support their long-term operation. So support the sustainability and capacity building on a European level. So this is a bit more in-depth view into the B1MG project that looks at the individual work packages. And I don't mean to go through this in any great detail. What I want to show you is that at the middle of this, and what a really important message from the B1MG project, is the citizens of Europe. So what we're trying to do is build a framework here that of course builds the federated infrastructure, which is a technical infrastructure that we see the need for from ELIXIR and many other places do. But at the heart of this is the need to improve the healthcare system across Europe for the citizens. This is a view of the same work packages, but now looking at the work packages into terms of what can be delivered. And again, I don't mean to go into this in any great detail. The slides are available for you to delve into after the presentation, or indeed, I can be on hand to have a discussion with any of you at a later date. But I've changed the word in the middle here from citizens to trust. because I think these are the two key things that the B1MG tries to focus on. So the citizens, in order to improve the healthcare for the citizens across Europe, but we can only do that if we can build trust across the partners. And as the first funded project to implement the European Declaration, this is something that's always in the mind of ELIXIR, to make sure that we are building trust, we are building relationships that are strong across Europe that allow us to have this pan-European infrastructure. To go back to the action that the B1MG project will deliver, ultimately it will be a capability maturity model. So we know that across Europe, many of the countries are at different levels of maturity in managing sensitive human data. So this figure of the capability maturity model, the vision is that any country should be able to self-assess where they are on this model, and the maturity model will explain in relatively concrete terms how a country can progress to the next level of maturity, ultimately really reaching this highest level of sustainability and the highest level of maturity. So what do I mean by that? So this is an example of a maturity level model. This is an illustrative example of what the B1MG project is trying to deliver. And again, we have these 5 key steps. We have initial, regional, national, European, optimized. And across the columns, we have security, infrastructure, personalized medicine delivery, and what the benefit is for the citizens. And so if we look at, say, the initial level, and we look at the security level, then the text there says that there is lack of alignment and regulation prevents sharing of personalized medicine data. For infrastructure, there is no national infrastructure available. For personalized medicine, it's impossible to link genomic and health data. And for citizens, no genomic data is taken into consideration on patient diagnostics. So we in this capability maturity model, start to work through the levels. We explain what the definition is at each level, the cost to the country for increasing levels. But ultimately, if we can reach this optimized level, then at the security, transnational access to data is available and it's machine ready. Federated cross-border infrastructure, again, is available, machine ready and based on common global standards, so that it's interoperable. Personalized medicine delivery, personalized medicine is delivered at a global scale, importantly, at a reduction of cost for the healthcare systems. And then for the citizens, ultimately the citizens, global access to these data allowed to tackle global issues and accelerate translation into healthcare systems. So the genomics data is being generated, globally available, and that's accelerating the transition of these data into medicine. So Andy, before we start on the GA4GH, which is a bit more technical, should we stop here for discussion and ask if there are any questions from the group?
We should, yeah, it's a good point. And Bogdan does have a question. So Bogdan, would you like to ask your question to Gary?
Yes. Hi, Gary. Hi again. So until now, a really amazing talk. And even if I was aware of some of ELIXIR's projects in the area, I'm discovering a new perspective. And to be frank, the vision that ELIXIR has is pretty amazing. And but going into a specific question, you mentioned there is a significant amount of data being generated and that will be generated in clinical settings in healthcare. And this data is useful also in healthcare, but will also be useful in research. I am aware of some projects that try to bring this data from healthcare into research. Was wondering if ELIXIR has any projects in this area to help bringing the data from healthcare to research, or so if there are projects, and what are your approaches, what ELIXIR's approach is? Thank you.
Yeah, excellent. So we, in a minute, we'll talk about the Global Alliance for Genomics and Health. But to answer that question right now, yes, ELIXIR is trying to expedite the transition of as much data from the healthcare system into the research system. And the way that we try and do that, or one example of the way that we do that, is to use what we call data use ontology codes. So as each data set is generated in the healthcare system, it can be tagged with a variety of different codes that describe at machine readable level how that data can be used. So one example would be a data use ontology code that can be attached to a data set that says this data can only be used in the research setting. Another data use ontology code would say this data is not able to leave national jurisdictions. But there are a variety of data use ontology codes that say things like was generated in the healthcare system, but can be used for researchers at the European level, can be used for projects that are non-profit making, so i.e. not industry, can be used by researchers that are working on particular phenotypes. So these are the types of codes that we try and have as many data, or at least the data use ontology is a strategy that we try and implore many of the ELIXIR nodes to have integrated in their healthcare system, which means as these data are generated, they are research ready. There's no need to go back and say, these are data sets that you generated X many months or X many years ago, who can use these data? The much more sustainable and high throughput way of doing that is to try and verify, is to try and tag the data set with the data use ontology at data generation. So if I'm in a healthcare setting, as soon as I've generated the data, what are the restrictions? How do I make those restrictions on these data machine readable? That's one key project. And the implementation of these data use ontology codes, although it's a standard that comes from GA4GH, is happening in many projects. One example would be the SUNICA project, which is an EU CAN project, working across 7 countries in Europe, Africa, and Canada. The data sets that are generated in the healthcare systems of H3 Africa, a huge project in Africa, CanDig in Canada, and also, I forget the name of one or two of the key projects within Europe. The data sets that are coming out of these essentially healthcare systems are tagged with data use ontology codes at the outset. Then at the machine readable level, we have an infrastructure called ELIXIR Authorization and Authentication Infrastructure, ELIXIR AAI, which can have me as a researcher associated with my credentials. We have the data sets across all of these projects tagged with their various codes, and right away I can see which data sets from these healthcare systems I can access with my authorizations. So I am a not-for-profit researcher. I've worked in diabetes in the past. Right away I can be tagged to data sets that have been generated in these healthcare systems that are focused on diabetes and available for not-for-profit researchers. This is the way to try and expedite this going forward and take us away maybe from the classical way to access these data, which is to ask the data generator with an email or a letter of intent. That's a bit slow progress. We have to try and increase the pace in the way that we do it is data use ontology codes.
So thank you, Gary. That was a good answer. And thanks for the question, Bogdan. When you resume, Gary, I'm going to have to ask you to go through at a little bit of a quicker pace, just so we don't, because you've still got the second part of your talk. And Bogdan, we'll come back to any questions at the end as well. I just don't want us to lose track of time too much. Thank you.
Yeah, sure. Andy, how long have I got left?
So if you can try and go through the next ones, the next part in say 5 to 7 minutes.
We can do that. Perfect. Shall I start now?
Yes, please. Okay,
then let's go through GA4GH. So many of you will be aware of GA4GH, and this is a global standard setting agency to simplify the way that people search for and request access to identifiable data in international and national genomic data resources. The way it works is to gather communities together that share a common interest. They present their requirements and build a technical standard that meets those requirements. The standard is driven through GA4GH and is approved as a product. And then as it comes out of GA4GH, it's driven back into the community and this drives the adoption of standards at a global level. So what I do is I like to add ELIXIR to this tag because we have a strategic partnership with GH4GH, and we've already gathered some of the communities together, as we've seen earlier, for federated human data, rare diseases, human copy number variation. And as the communities build their technical standards, again, we can drive them through GH4GH, and then when they come out, we can use ELIXIR and the ELIXIR instruments to increase the uptake of these standards within the communities. So really the objective here is to generate standards from the community and then increase the uptake of those standards within the community. So we have formed an ELIXIR GH4GH strategic partnership. This means that we have now a methodology for the ELIXIR nodes to speak directly to GH4GH leadership, to present their requirements and present what we need at a pan-European level from the standard setting agency. And if we look across the ELIXIR nodes, the majority of ELIXIR nodes contribute to this strategic partnership. And it allows us to find a way to speak directly to the leadership of this global standard setting agency and speak with one voice to say, what are the needs of European research infrastructure? Ultimately, it's a two-way partnership. Although ELIXIR gets to try and have as much influence on GH4GH as possible, GH4GH is able to use ELIXIR for the dissemination and implementation of the standards at the European level. So I'll now go into two examples of where we've used our relationship with GH4GH to build a product and build products that are in use across Europe. And the first is in data discoverability. So in our figure of this data federation framework, how do we find data sets that are across the globe in a standardized way? If you have data in these sensitive data archives, how can I access it from wherever I am? And the product here is called ELIXIR Beacon. So ELIXIR Beacon is a standardized API technology that allows the discoverability of genetic variation data. So it's relatively simple, that allows one to ask, any researcher to ask the question, do you have information about this allele at this position on the human genome, chromosome 13 in this case? And the beacon replies with yes or no. So at a global network level, we're able to say, if you have variation data and you light a beacon on that variation data, you can allow this data to be discoverable by anyone across the globe in a network manner, in a federated manner. So the data never has to leave where you are, but it can be discovered. And then what we also do is integrate this ELIXIR Beacon with something we call the ELIXIR AAI to provide access at 3 different levels. So not only can you access the data at the public level, which means anyone with internet access can ask the question of, do you have this allele at this position? And you can respond with yes or no. But if you've logged in with ELIXIR AAI, the beacon owner will know more about you. So the beacon owner in this case will be able to say, this person here that logged in with ELIXIR AAI, do I want them to respond? Do I want them to query my beacon? And if they respond with yes, they can give more information. The same is true about the controlled level of access, which means we can use the ELIXIR Beacon to ultimately provide answers to federated data discoverability. So I've gone through that relatively quickly, but at the bottom here, there is a link to a YouTube video, which will take you through that in much more detail, or indeed, please do reach out to me if this is of interest to you. So the next thing I'd like to talk about is, and lead into the next presentation from Kathy, is that of the COVID-19 use case. And that's a current driving use case across Europe for this federated infrastructure for cross-border access to sensitive human data. And and our key ELIXIR product here is that of the EGA, which is an ELIXIR core data resource. So the EGA currently works on a centralized model where it is based in the EBI in Cambridge and the CRG in Barcelona. But the EGA, more necessarily, is moving to this federated EGA model. Because like we said earlier, where we have these sensitive data archives, we know it's no longer possible in many cases for the data to be submitted, the raw data to be submitted to these central archives, it must remain where it is. But we have to find a framework that allows us to access these data transnationally. So what we can do is share the metadata to the central EGA, which means data sets can still be discoverable, but the raw data remains housed in these secure, sensitive archives. And as we work towards this federated EGA model, there are 3 levels: the central EGA, EBI in Cambridge and CRG in Barcelona, federated EGA nodes, the sensitive data archives across many different European countries, and the EGA community, which are able to consume these data and analyze these data in clouds. The COVID-19 has been a key driver of this use case in the last months, ultimately leading to the European Commission offering ELIXIR the opportunity to extend one of our ongoing projects, which is ELIXIR Converge, with a new work package to essentially accelerate the implementation of the Federated European Genome Phenome Archive. So we know that there's a need for the rapid sharing of human cohort data for this COVID-19 pandemic response. And the objective of this work package 7 within Converge, which has many of the ELIXIR nodes resourced within, is to support the underlying architecture and the technical interfaces that are needed for the federated EGA, establish software implementations for nodes, and then also, again, we come back to this maturity model level idea. So we should, within this work package 7, provide a node maturity model. So I'll allow any ELIXIR node to say, where am I in my capability of hosting a federated EGA node? And then how would I move to the next level? And ultimately, the aim is to build this European COVID-19 data portal, which many of you may be aware of already. And I'm sure that Kathy will come back to in the next presentation. So the summary here is that the Federation is the solution in the age of healthcare genomics data democratization. ELIXIR Human Genomics Translational Data coordinates the implementation of a federated network to manage sensitive human data at population scale. The majority of ELIXIR nodes are involved in funding and HGTD activities. All ELIXIR members are welcome to join, and indeed there's an opportunity for non-ELIXIR members to join in many of our activities, and I'd be interested to talk with anyone that has a vision there. We work with the EU and other funding bodies to align opportunities. For example, the 1+ Million Genomes Declaration. We're a strategic partner of GH4GH, and I've presented the technical solutions of the ELIXIR Beacon and the federated EGA. So I'll give my thanks to all of us that work within the ELIXIR nodes and wider, and hopefully we have time for any questions, and if not, hope maybe you can reach out to me via email or the discussion part later this morning. Andy?
Thanks a lot for that, Gary. It's really comprehensive. So I think we will take 1 or 2 questions. I've seen that we have a question from Anka Kostash, and I'll read that out to you, Gary, so that you can answer that. And then we have a question from Bogdan as well. So Anka wants to know, so you mentioned that the vision for ELIXIR needs to be translated into national regulations.
Yep.
So can you explain how this process worked for other countries?
Sorry, Andy, you broke up at the last bit there. I had vision.
Her question, that you talked about the vision ELIXIR has in the area of human data. The vision needs to be translated into national regulations. How did this process work for other countries?
Yeah, that's a key question. So I think we lead by, so how it worked for different countries is different in pretty much every country. But what we try and do is work with projects where we can bring the key people around the table to discuss the requirements. So one of these projects is called EU stands for PM. So this is, EU stands for personalized medicine. Within this EU stands for PM project, which is focused on a lot of the regulatory and ethical issues, we ask for examples of how this has worked within one country, and we describe this to others. This is also something that's happening within B1MG. So we have something called lead countries, and they would be the ones you would expect. So perhaps the UK. How did the UK go from having Genomics England, which is now a national infrastructure, a national healthcare focused genomics data generating project, to something that can be shared transnationally. It's a driver project of the GA4GH. You can access these data in different places. How did that process work? So in the case of B1MG, and in the case of EU stands for PM, we have a country visit. We get a delegation that goes to the country, it asks key questions, and it brings that information back to EU stands for PM. So there are a variety. So if you look at specific examples of how it actually works in those countries, it changes in very many places. What's happened within England and the UK is very different to what's happening in France, for instance, where data are held at different levels of security. But what we try and present to the ELIXIR nodes or the partners in these projects, like EU stands for PM, is the way that they did it. And if we can present it the way that they did it, or from more than one country, then it allows other countries to say, okay, that's perhaps something we can adopt, maybe something from France, something from the UK, to try and build the capacity at a European level.
Okay, thank you, Gary. We have one question from Bogdan as well.
Sorry, yes, I can ask it. So I'll just read it. Currently, the gene panels represent the majority of the data that is generated in health, and that in Romania, and I don't think it's only in Romania. It's not the exome, it's not genome data, but this is still very valuable data. How would we integrate it?
Yep. So I would, thanks for the question, key question. I completely agree that gene panels are the majority of way that we're seeing these data generated right now. That might change in 3 to 5 years, but it is true that that's the case across many European countries. I can't see you and I'm not exactly sure who asked the question, but for our answer right now, I would ask you to look at the panel app product of GA4GH. So this is something that's been generated by Genomics England and is now in collaboration with Australian Genomics, something that's been adopted in many places that standardizes the way that we can look at panel data, map it to what we call the GenCode gene set, and then look at the interpretation of those variants. So panel app is something that we adopt within ELIXIR, within our GA4GH strategic partnership. And ultimately, it's the way that we try and standardize these data. I synonymously use panel data the same as I use genomics and exomic data, because there is a framework to map these data to all different aspects of the GA4GH suite of products.
Excellent. Thank you, Gary. That was great. Okay, so we were at the time where we were going to have a break, but I think what we will do is continue with Kathy's talk and then have the break after that. So if I can invite Kathy now, who's ELIXIR's industry officer, but also covers the portfolio of coronavirus activities and services in ELIXIR. So Kathy, if you could give your presentation now, please, that would be brilliant.
Thank you, Andy, for the introduction. My name is Kathy Lauer, and I am going to tell you a little bit more about our COVID-19 initiative, which was only established this year. So very early in March, we when everyone went into lockdown, we started thinking about what we from ELIXIR could do to support the research community together with our members in fighting COVID-19 disease. And the first thing we did was to have a look at our services and really compile a list and a comprehensive overview of the services that are already available through our members that are specific to COVID-19 research. So I would invite everyone on the call to go to our website and check out this COVID-19 specific site, because there is so much information on there and I don't have enough time to walk you through everything. But the services that are offered are really wide and broad and very of different kind, and can range from databases to workflows, etc. So this was the first thing I wanted to point out, and just to show you what we have done to really bring in the research community, make them aware of what is going on in ELIXIR in regards to COVID-19 work. We have another section on our homepage that I would invite you to check out. This is the news section. here you can also in the future read more about success stories and highlights around COVID-19 related stories. And one of them I want to point out, this was one of our latest stories, was on the COVID-19 viral beacon. And Gary has already spoken about the beacon protocol. So researchers who had been working on the beacon protocol for a while were able to quickly repurpose this resource and make it available for researchers to browse SARS-CoV-2 variability in the genome of the virus. So yeah, these are just two things to start off my talk, and they will give you a lot more information than what I can give you today in these couple of minutes. But so one thing I want to point out before I give you more specific information is to answer the question, why does sustained investment in research infrastructures pay off? And with COVID-19, we have really had a case study that put an emphasis of why this is so important and what we can achieve when we really invest long-term in a network of experts and infrastructure. So I want to explain that on the example of the COVID-19 data portal, which we've been collaborating on with the European Bioinformatics Institute and many other national members. So many of them are ELIXIR members across the European continent and with the European Commission as well. So as you all know, and as Gary has already pointed out, at the beginning of the coronavirus crisis, a lot of people kicked into action, a lot of research centers, a lot of universities, public health laboratories, hospitals, medical centers, and started producing a huge amount of new data in relation to the disease and the virus. And then, of course, this data had to be shared and had to be put out into the public. So for the data there are, of course, a huge amount of diverse resources that can be used, and many of them are really disconnected and scattered. So I've just pasted a couple of resources that might be quite familiar to you on the slide, and these are resources that every researcher probably knows. Okay, and of course, then following on from the research generation, it was important to make sense of this newly generated COVID-19 data to get more insights on the virus behavior and of course the implications on human health. And to kind of combat the disease, it was of course important to bring all those three layers together. So very early on, when we went into lockdown in March, we started working on bringing all those resources together. And this looks like a quite complicated slide, but I'm just going to walk you through it. So in the top layer, you can again see all the data generators that were generating either viral data or human clinical data. And of course, all those data generators were looking to deposit their data. And ELIXIR, or our members, have already a huge variety of databases available. They've been established over many, many years, and we're able to really hold a huge amount of this data and help people to make their generated data available. So this can either be access controlled data or open research data. And this was all brought together in the COVID-19 data portal, which is hosted by the European Bioinformatics Institute at the moment. So I've already touched on that, but we were really able within a couple of weeks to put up a platform that helped to flexibly take up this generated research data and made it available and connected on this platform. And this was really we were only able to do that really because we could repurpose resources that had been developed within our network for a while. So as you can imagine, you cannot set up something like that overnight. So we really were able to build on our work of the last years and really react very fast when the COVID-19 use case came up. So as I mentioned before, we were building on an infrastructure, and Gary had touched on this before. This can either be access controlled data or open research data. This data can go into one of our many established databases that hold anything from genomic sequences, protein sequences, expression data sets, and really bring them together and interconnect them. And of course, one aspect that was really important in that respect, and I think this is one of the many added values that being a member in ELIXIR and participating brings, is this network of people and people who've worked together for many, many years and are used to working with each other and can be drawn upon very quickly in a situation like a pandemic. And of course, this kind of leads on from the people aspect, which is trust. So we've worked with ELIXIR members, with the European Commission, with the European Bioinformatics Institute for many, many years. People know each other, people have worked together in the past. There is a lot of trust to move forward quickly. And of course, as Gary has already mentioned, this work is not only focused on COVID-19. COVID-19 really has been a use case for what is to come in the future, and that is to develop Europe's future health data landscape. And we're already involved in many, many projects that will lead the way and really, really will help to lead into that federated data sharing across borders. And I think the COVID-19 use case has really helped us to show what is already there and where we still need to put a lot of effort and work in to make this reality. And some of the projects that have already been mentioned that will take that further are, for example, ELIXIR Converge, the European Open Science Cloud, or the 1 million genome Beyond 1 million Genomes project. So just my last slide here. Yeah, this is just to summarize that the COVID-19 use case is by no means will end with the end of the pandemic, but will be taken further and really expanded into other areas of human health as well in the future. So yeah, this was all from my side. And if there are any questions, please reach out to me. Andy, you're on mute.
Thank you, Kathy. Yeah, I was just saying it was a great talk and it was a good summary to of our work in this area and how it fits into the health space as well. So thanks for that. I can't see any questions at the moment, or I also can't see any raised hands as well. So, but maybe I think I would like to come back to a point you mentioned, which is that at the moment, research groups from anywhere can also put their data into the COVID-19 data portal. And as I understand it, there are regular meetings through the EBI and through ELIXIR with representatives of national communities, aren't there? So I wonder if some of the delegates, some of the attendees on the call wanted to mention work that they're doing to put into the COVID-19 data portal, or Kathy, if you had any advice about what they should do. Is there somebody that they need to be in contact with if they need to deposit data in the portal?
Yeah, so I mean, in general, I can just say, if anyone has any questions, please feel free to reach out to me. Or if you go directly to the website of the data portal, then you will find the respective contacts there. I will post the link to the data portal also in the chat box, so you can have a look at that.
Thank you, Kathy. Bogdan?
If I may add on that, maybe I mentioned the last meeting, but there are 2 Romanian groups in contact now with EBI, and these groups will deposit the data on the European portal. The data was generated and is deposited on GSAID, but now these groups are working towards depositing in the European system. And I think that's a good achievement, and we'll let everybody know when this happens.
Okay, great. Thank you, Bogdan. Okay, I'm going to pause the first part of the webinar now so that we can have a chance to refill coffee cups and so on. So it's now 15 minutes past. If everyone can be ready to restart again at 25 minutes past. Okay, so 25 past, we'll restart the webinar. Let me just very briefly introduce Hedi when you're getting your slides set up. So Hedi, we're really delighted that you could join. She's going to be giving a presentation about ELIXIR Estonia and giving examples of some of the human data related activities that they lead and that they're involved in. And Hedi herself is Deputy Head of Node and has been involved in ELIXIR Estonia, been one of the main driving forces behind it since the beginning. So it's really great that you can give us your perspective about some of your human data related activities.
So hello everybody. I think we have met with some people already, and I had a very nice meeting with Bogdan in Romania a few years ago. So I'll talk about 2 things that somehow connect to each other, about ELIXIR Estonia and our services and projects, and then what Estonia does in human genomics. Estonia is a small country. We are the similar size to Netherlands or Switzerland. So I come from University of Tartu at the southeast part of Estonia, and our other partners are Tallinn Technical University and the National Institute of Chemical and Physical Biology in Tallinn. So it's a 2-hour drive, and our main airport is in Tallinn. So every time I go to some ELIXIR meetings, I take the drive. So we have 3 member institutions, as I said. Our head of node is Professor Jakvila. So I'm coordinating all the daily actions. We do have excellent board members and scientific advisory board, both local and international people involved. Running the ELIXIR Estonia, it's a small team. We have Ulvi, who is working mostly on the human data. Priit, who is also a scientist working on different aspects. Then we have a developer team of 3 people. And we have also a dedicated person from the computing center who is helping us with all the maintenance of the software. So Jak, me, and Martin, who is taking care of all the paperwork. So small and effective team. So our key interest, goes beyond ELIXIR Estonia key interest, but it's, I would say, Estonian interests and in general, like the bioinformatics and health science interest in Estonia. So we provide e-health electronic health record analysis for the Estonian Genome Center, genetic risk assessment, pharmacogenomics analysis, omics integration using public data and Genome Center data, causal effect analysis, EQTL analysis. I will touch some of these topics later on my slides. And then we have a big interest in machine learning and AI, especially image analysis, and our people are part of the ELIXIR machine learning team as well, or the focus group. So about ELIXIR Estonia interests, so we have 2 main areas that we contribute to. One is tool development. So some of these tools are part of the Estonian service delivery plan. So the function enrichment analysis tool, that's I think the most famous one, is a G-profiler. But also ClustFees that we just celebrated yesterday of 1,000 citations. It's a tool for visualizing clustering of multivariate data. And some other tools that might be useful for for bioinformaticians or the biologists at labs. And then our second goal is to train people on these tools and services. And the training also goes beyond our own services. We have software and data carpentry instructors and also people who have taken the ELIXIR Train the Trainer courses. So they have been trained to train adults. We are happy always to train basic and advanced R, Python, command line, Git, and so on, but also functional interpretation, data visualization, and machine learning. And some of these things our people are training at ELIXIR courses, for example. And potential contribution that you might want to contact us about are if you want training on the tools or services of ELIXIR Estonia, carpentry trainings, something on machine learning or personalized medicine. And we are also happy to host people in our research group. We just moved to an awesome new building at the beginning of the year, and then we went home 2 months later, but now we are back. And now switching gears and going to the human genome, more into that. So Estonia has one of the first biobanks, I think, that was established in Europe. It's a prospective longitudinal volunteer-based biobank. We have 200,000 participants, which means it's 20% of Estonian adult population. It's quite a lot. And there's different type of data collected. There are health records, diet, physical activity, and other questions that the people answer when they join. Then there are DNA, plasma, and cell samples kept in the liquid nitrogen. What makes Estonian Biobank special is that there is Estonian Human Genes Research Act that was written up very early on, like now close to 20 years ago. The people signing up have broad informed consent, so they give the right to do a lot of research, and it's open for research. There's clear access rules, and the Estonian Genome Bank has had very many collaborations. So how the data, one of the screenshots of the questionnaire in coming slide, but there is personal data, then there is data about how the genealogy, about the parents and siblings and children and their diseases if known, the health behavior, how active people are, what are their smoking or drinking habits, the disease information, and the objective data, the blood pressure, waist, the hip measurements, and so on. So this is how the way the data is saved. It's a fairly complicated form. This is not filled in by people themselves, but it's and this is actually an example of tuberculosis registry that's connected to the biobank, but the biobank questionnaire is very similar, and it's a specialist who asks the questions and fills in the forms. So another thing that makes Estonian Biobank special is the way that it's linked to other registries in Estonia. So Biobank started to have the first people joining in 2002. And then since 2010, there is linking to different registries, like population registry, which tells us where people live. Death registry, so this is the cause of death and the date if a person has passed. Then we have the cancer registry, tuberculosis registry, we have HIV registry linked, then two of the major hospitals, the North Estonian one and the University Hospital in Tartu, they link their medical records. Then there is the drug database and health insurance funds information that are also linked. So this all comes back to the genome bank. And nothing would happen without the IT. So I would say that the systems that were made to connect Estonian databases or make Estonian citizens digital have played a huge role for the genome bank as well. So we have the ID code and electronic identity. So the ID card, that's a picture here, came to play on 2002. And it's a mandatory thing to have everyone. Sorry for that. And this is you can't see well, but there's a reader that you can log in with. Then 2007 came mobile ID, something that I use a lot. So I just need my mobile with me, and I can sign digital documents and authenticate myself. And Smart ID app came a few years back. But the thing that's behind that is X-Road infrastructure that links to all the information systems in Estonia. So there is logging, timestamping, and so on capabilities. But regarding to the genome bank, as I mentioned, there is the consent form for the biobank. Then there's nationwide billing information. So all the procedures that are done to the people are billed, and these are saved and linked back to the genome bank. We do have the electronic health records, the medical notes that the doctors do, because they have to upload all of these to the central database. Then we have 99% of the drug prescriptions are digital only nowadays. And then we have the national health registries that I mentioned, and these all link to through the X-Road infrastructure. So this is another way to look at that. That different databases are put together to make the data about the gene donors as descriptive as possible, and allow a lot of different research that's not possible in many other countries, for example. So switching again a bit to health data analysis. There's a lot of text mining that we do because we speak Estonian, we write Estonian, so we can't really take programs just from the other large countries applied to our own language. It's a fairly tricky language to learn, so a lot of effort has been put into that to make for example, the electronic health records that involve a lot of free text readable. So machine learning is a central point to predict the context, to the semantic meaning, semantic similarities, to extract the information from the unstructured data. Very often even the blood analysis are unstructured. Then to do disease comorbidities. And trajectories analysis, quality indicators to compare observational data and registries. And for example, we have been part of the EMI-EMIF platform doing converting the genome bank data to the OMAP format. And a lot of effort has been from our bioinformatics community been to link the genomics data to electronic health records and create the needed information architectures. So a few of the examples that we have done, for example, medical care spendings. So this is a plot for a few years back, but on the x-axis are the age of the people, and on the y-axis are patients per one inhabitant. It's and we are looking at the costs across different diseases. Each of the disease groups are on the right side and by different colors. And in 2016, close to 600 million was the health costs in Estonia. And we can see that there's a peak around the 30-year-olds, which is and very wide peak of the pink. So this is usually when people get kids. So it's the mother and kid costs in hospitals, delivery costs. And then we can see that the health costs start to rise after people get 50-year-old, and then they drop after 80 because we don't have many people living beyond 80. And we could zoom into some of the disease groups. I have highlighted here the circulatory system diseases, so different hypertension or rheumatic heart disease and so on. And this allows, for example, the Estonian Health Insurance Fund to look at where the costs are and where the money actually goes, and to take actions based on that. Somehow related to that, but another twist of what the Estonian Genome Bank does, and along with our bioinformaticians, is pharmacogenetics. So they translated the genotype data of 45,000 people to actually come up with recommendations and find out what are the challenges and solutions there. So in our genetic background defines how well we can, how well do we metabolize drugs. And there are some very well-known alleles in our genome. And then this our phenotype defines how good or bad we are metabolizing certain drugs. And we can see, for example, on this top right panel, that there is like 1/3 of the people who are normal metabolizers. There is like 1/4 who are intermediate metabolizers, and then 1/3 who are rapid metabolizers based on this particular phenotype. And if all of these people are subscribed the same dosage of the drug, then for some of it, it's totally fine. For some of the people, the dosage might be too high because they metabolize it not as fast as the normal is. And then for some people, they say like the drug doesn't work because they metabolize it so quickly that it can't have its effect. And for other locations in the genome, the ratios between these groups are different. And why does it matter? As I said, for some allele and genotype, you can take drugs and you don't have to care what's the dosage. But for some drugs, you really should know that you have a much lower dosage or much higher dosage. So this is one of the examples that the Estonian Genome Bank provides back to their gene donors. The example is about a 33-year-old female with depression. Apparently, she's a slow metabolizer based on one of the SNPs, and she's recommended 50% dose reduction because of that. And due to the Estonian Genome Bank having the data of their electronic health records and drug prescriptions, they have seen that that person was subscribed previously by sertraline and citalopram that are affected by this particular SNP or genotype. And both of these drugs were discontinued due to side effects. So she actually got overdosed. And but if she would have taken genotyping before going to the doctor, or the doctor would offer or order a genotyping, they would know that probably either, if possible, avoid these drugs or give much lower dosage. So this is one of the benefits that Estonian Genome Bank participants now can get back. Another thing that where the Genome Bank went back to the donors was familial hypercholesterolemia project. So they screened 50,000 people for hypercholesterolemia. Didn't find too many people, but still a few tens. And they studied the cascade or family members as well. And in total, they found 41 carriers with 11 distinct variants. So 20 family members from 16 families. And they called all of these people back, offered them clinical examination and genetic counseling with cardiologists, and did the risk stratification. So they made these people aware that their genetic background might lead to cardiovascular diseases much earlier than they would maybe expect. And thus the people can react early on and hopefully survive for a much longer time with a good health. So this
Hedi, sorry, Hedi, I'm just to remind you of the time. I'm not sure how many slides you have left, but
I'm at close to the end.
Thanks, Hedi.
Yes. So this is one of the examples again of the feedback that the people will get, that they have their data, then what's their diabetes risk? That's a low for that particular person, but has a high risk for the cardiovascular diseases, has a genetic risk of early menopause, for example. And for diabetes, they gave like, if your weight is on this level, then your risk is that high, or if you would gain weight, then what would happen to your risk? So this is the architecture behind that. So there's different tools to harmonize, consolidate the data. We have genomic data, phenotypic data. Then the X-Road connects all the external databases. And then there is the pseudonymization and research and development tools. Of course, plenty of challenges lying ahead. I would not stop there. Just to mention two more things. Estonian ELIXIR also set up Estonian Beacon. So this involves the full genomes of the Estonian Genome Bank, the indels and SNPs, and it's based on Illumina VGS data. And that's available from beacon.ut and also from the central Beacon website. And as COVID was mentioned, ELIXIR Estonia almost dropped everything that was on our plates in March, and we also set up a few of the tools, launched a questionnaire or voluntary survey to collect information, and these are available at the corona.ut.ee. And it's a huge team of people, like huge team for Estonia, probably a very small team for the international manner, but there's a people at the Genome Center who are leading these efforts. There's the Competence Center team, the Scientific Computing Infrastructure team, then our bioinformatics group leaders or young scientists there. And all of that is one way or the other connected by Jaak Vilo, who is also head of the node of ELIXIR Estonia. So I'll stop here and happy.
Thank you, Hedi. And so we will take just one question now, which we already have on chat. So I'll read that out to you just to save some time, and then we'll go on to the next presentation and then a discussion at the end. So the question from Bogdan: one of the main problems to reach such a high number of federated genomes is convincing participating labs to share data. Was ELIXIR Estonia involved in this process? And if so, can you share some insights?
So I guess you mean the 1 million genomes thing, because Estonia is part of the 1 million genomes. And I think for the very beginning was clear that Estonian genomes that we have, or we're going to produce more, will be part of that. The 2,500 full genomes that we already have, I think this was not the problem. The problem was money. Estonian government at that point gave money and the sequencing was done abroad, but there's no constant flow, so we can't do full genomes of all the 200,000, unfortunately.
So, hi, Hedi. Just to come back to my question, were these genomes sequenced centrally, or were they generated at different labs and then brought together?
No, these were all sequenced due to the people being part of the Estonian Genome Bank. So the Estonian Genome Bank got money to sequence some of the people. So because we wanted the Estonian reference genome to create the Illumina chip that picks up the Estonian specific SNPs. So that was the initial goal. So I think there are other labs in the medical faculty as well that do some of the sequencing, and we are setting up the local EGA that should collect all of these in the I would say hopefully starting from next year, because there is a huge interest, but it takes a bit to set it up.
Okay, thank you.
Excellent, thank you, Hedi. Okay, so our last talk is from Cristian Zimbru from the Center for Genomic Medicine in Timisoara. And Cristian will give his perspective from a Romanian human data research lab. So over to you, Cristian. Thank you.
Yeah, thank you, Andy. Thank you for the invitation. My name is Cristian Zimbru. I'm an assistant professor at the Polytechnic University Timisoara, and also a bioinformatician in the Center of Genomic Medicine. So in this presentation, I'm going to present some insights from the Center of Genomic Medicine, the objectives, the equipment that we have in the center, the team and the projects that we did, and of course some information about our bioinformatics facility. So the Center of Genomic Medicine was built in 2015, between 2015 and 2016. We started somewhere in the, operating somewhere in the summer of 2016. The main objectives of this center was to provide infrastructure for biomedical research, also train scientists in the field of genomics, and overall to create a center of excellence at European standards. So we have in the lab, we have like two sequencing machines, like an HiSeq and MiSeq, two microarrays, and a lot of other devices that can be found on the ARIS platform. In the bioinformatics facility, we have 4 servers that total up around 80 terabytes of storage, about 150 cores, and 800 gigabytes of RAM. So the team in the lab, we have seniors, which is Maria Puiu, who's the director of the lab, Nicola Andreescu, who's the NGS lead. We have Simona Farkas, who's the lead on cytogenetics, and we have Adela Chiriță, who's the lead on clinical interpretation. And of course, we have also some amazing PhD students and postdocs. So what the main analysis types that we perform there are DNA sequencing, SNP array, and karyotyping. Of course, we have the appropriated bioinformatics pipelines for these analysis types. So we can do secondary analysis, which is generating the VCI file from the FASTQ. And we also have the tertiary analysis, which consists of annotation and filtration and prioritization of the variants. We started recently looking at the copy number variation analysis, and also at the RNA-seq analysis, because we started doing whole genome sequencing, and for the whole genome and whole exome sequencing. And we had a request for RNA-seq. So in the bioinformatics facility until now, we have processed like 1,500 subjects. These patients come from all over Romania. And of course, they come to us, but they are also sent by our external collaborators. I just have here a very short list of these collaborators, but there are many others. Also, we had a project that just ended in March, where we sequenced 600 subjects, and we collected around 400 data points about these subjects. The results of this project was a patent application and a few research publications. Of course, the data is still being analyzed. Other projects that we were involved in and we are involved in. Of course, in March, our team was called up because we had the expertise and the equipment for testing for the SARS-CoV-2 or the coronavirus. We have also an ongoing project which is for rare cancers, and in this project we have 50 subjects on which we will perform whole genome sequencing, and we got another project that's funded by the Horizon 2020 initiative on the obesity policy. So I will just keep it here very fast. What we are looking to do in our bioinformatics infrastructures is constantly update the tools that we use and the pipelines. Now we have like all the pipelines are in virtual machines, so we are looking moving them to Docker. We also have generated the SNP frequency for our patients, and we still have we can access this data structure, but we need a bioinformatician for this. So we are looking to create a platform where we can, I mean, any of my colleagues can access the data without specifically asking me to do that. So about the data management in the center is composed of mainly of two data sets. The first data set is stored on the servers and consists of sequencing data, I mean the FASTQ, the BAM files, the VCF, the annotation, and we have also the clinical data that is stored on a different machine. We are, I just mentioned that we are looking for a platform that can integrate this data locally. We have, of course, we had some data profile. When we try to interrogate the data, we had some issues. Also, we have some issues with the storage management, because some of our virtual machine got really big and we had to move data around. So how do we interact with the national databases? Well, we do not interact with the national databases because they are missing. We do not have like SNP frequency in the Romanian population, or we do not have data sets that consist of phenotype and genotype bundles. What are the reasons for this? Well, the first reason I think is the legal reason. The legal reason, yeah. I don't know who has. We don't know who has the rights to share this data. So is the patient, the laboratory, the ministry? The infrastructure is missing. There is no effort for collecting this data, and to some extent, I believe there is also an attitude which is if a doctor has the data for a patient, it would not share it with others. So are we alone? Are we the only NGS laboratory in Romania? Definitely not. There are 6 regional centers by law, and there are more than 10 platforms in Romania for sure that do NGS. We do not know if they all do whole genome or whole exome sequencing, but maybe they do just small genes or small panels, but they are there. So I think national infrastructure is quite necessary for all these centers. So how do we interact with the international databases? Well, usually we use data aggregators like ANOVAR and Web, where we extract the SNP frequency or information about the genes from ClinVar or MIM, and some in silico predictors. Also for the SNP array interpretation, we use Decipher. Do we share data with Beacon or EGA or ENA or any other data repository? Of course not, because I just mentioned the legal issue. Do I think that the usage of these databases is relevant for the clinical interpretation? The clinical interpretation, of course. I think it adds another dimension to the process. So for the national infrastructure, I think Gary can help us here with some suggestions. So solving the legal issue, I think, is the first priority. Then, of course, collaborating all the way. And of course, we don't want to stay in a bubble in Romania. So we better connect with the international initiatives in the field of bioinformatics and others, other field. Just to prove the concept of collaboration here in Timisoara, we have different groups collaborating for the training of bioinformatics. And this initiative was started in 2017, and it's still working today. An extension to this initiative was the RSBI seminar that we had here. So I will end my presentation on this note that we need to collaborate and get things going. Thank you. I try to be very short in my presentation.
Thank you, Cristian. But even if it was short, it was very thorough, and it was really nice to see some of the projects that you're working on as well. That was great. Okay, so we're gonna, before we finish the session, there's a chance now to ask questions to Cristian or to any of the panel members as well. So as we've been doing throughout, you can add a question to the chat if you want, or you can raise a hand, and that will allow us to invite you to ask the question directly to the panelist. If you add the question to chat, then I will read it out to the panel members. So that's the process that we'll use for the next 5 or 10 minutes. So thank you, Cristian. Just when we're waiting for some questions to come in, you mentioned that you might like to get Gary's perspective about how you can connect some of the work that you're doing in ELIXIR to some of the European activities. So maybe it's a chance for Gary to reflect on some of that and give some suggestions.
Sure, absolutely. So I think with the current status of Romania, there are two key possibilities. The first is through the Federated Human Data Community of ELIXIR. So within ELIXIR, we have two relationships to communities. One is that of community member, open only to ELIXIR members, so that wouldn't be possible. But the second is that of community partner. So the community partners can join the monthly meetings. They have access to all the documents, and they can align with some of the deliverables that we work towards. And the discussion within Federated Human Data right now is to say what are the standards that we need for the infrastructure to work at a pan-European level. So I think from watching your presentation and listening to where you are, Cristian, with the mention of EGA, the mention of Decipher and other things, this would be a nice place to start. The second place is within GH4GH, which is a global alliance for genomics and health. Now this is a global standard setting agency, and there's a lot of work going on there. But I bring it up now at a pertinent time because today there is the annual meeting of GH4GH for today and tomorrow, which is happening online. And it's free for anyone to join. It starts this afternoon and it runs through to tomorrow afternoon. And for anyone that's thinking of getting involved in GH4GH or interested to see what's happening there for the standards at an international level, that might be a nice place to watch a couple of presentations if you have some time, or catch the recordings later this week to see what's happening there. Because ELIXIR, as I tried to explain in my presentation earlier, we rely on that partnership with GH4GH to really develop and then implement the standards for sharing these data at an international level.
So can you share the link to this meeting? I guess it's an online meeting, or yeah?
Absolutely. I'll put it into the chat window now. It looks like someone might have already done this. Yeah, I think Corinne's pasted it already. That's great coordination. Thanks, Corinne.
Yeah. Okay. Thank you. Thank you.
Bogdan, did you want to say something about maybe anything you want to think about nationally, how to coordinate these activities going forward?
So in fact, I would like to go down from nationally to a lab level, because sometimes things are difficult to drive nationally. And what is enough is that a lab take the initiative and showcases how the interaction can be done. And I asked a question on the chat, and I have the same question. Gary, and in fact, all the other panelists, you mentioned some tools, some structures, some data structures that like Beacon or EGA. can they be implemented only at a lab level, supposing that there will not be a national consensus? Can a lab, for example, use Beacon even if the country doesn't take any initiative in this direction? Can a lab have a local EGA?
Yep. So as a straight answer, and then I'll give more information, the answer is yes. So an individual lab can light a beacon on their variant data and connect it to the network, and then this data can be searchable in a federated manner. For an EGA, a lab can have a local EGA, store their sensitive data securely, and then share it on an international scale or whoever they want to share it with.
Yeah, I would.
The way that it can work national, I'll come back to you just one sec, Hedi. The way it could work to relate to the national infrastructure, I'll use one example, which is that of ELIXIR Spain. So ELIXIR Spain is coordinated at the national level by BSC, the Barcelona Supercomputing Center based in Barcelona. And the head of node there is Alfonso Valencia, and the technical coordinator is a guy called Salvador Capella. Now, what they do at a national level is work with the individual regions of Spain. And one of the key projects they've got going at the minute is called TransBioNet. So Salva has worked at with 14 individual clinical labs across Spain to develop this network of clinical labs that want to try and share their data through ELIXIR Spain. So that's the way that the different levels can work. So, okay, we see the international, so we see the national coordination from Salva, that comes into ELIXIR, but at the national level, Salva, as the technical coordinator of the Spanish node, is working with the individual clinical laboratories, and the individual clinical laboratories are the ones that are sharing the data directly. So they have the beacon lit, they have the local EGA, they have the technical infrastructure, which Salva starts with them and then starts to build up the way. Hedi?
Yeah, I would like to comment that for a larger country, the activities in Estonia, what we do might be like one lab doing something, because Estonian Genome Bank in a way is one lab. So we have lit up the beacon for them, but it's in reality, like most of the work was done by the computing infrastructure that is just that is supporting the Genome Bank anyway. And it's linked to ELIXIR because we are just working at the same institute. So I would say definitely, yes, all of these things can be taken up by individual lab if there is a will. And I think if you're afraid that it will be hard to coordinate at the national level, then I think it might be even better to start from the bottom up and show examples, and then the others want to join in.
Yeah, that's a very good point. Yeah, that's an excellent point.
Yeah, excellent. So I've noticed that we have a question from Raul Cristian. Please, can you unmute yourself and ask your question, please?
Hello, I hope you're hearing me well.
Yes.
So I'm a doctor in Romania. I'm very enthusiastic about hearing there was in Estonia, there was something about a volunteer data bank of genomes. I know there are some places around the world where the governments attribute the biological data of people to themselves. So I envision if such a global network of genomic data is being gathered from the people, I'm thinking me as a member of the public, can I express my will to share my information with this network?
So I think it's a very good point and question. So in Estonia, as far as I know, like in principle, person could ask their full genome from the Genome Bank. I don't know if anybody has done that, to be honest. But and I don't really know where would that person, like, they could probably submit it to the EGA, but that's, I think it's not that easy for a single person to do. So it's a tricky question, what a single person with their private genome, how can they contribute to science, for example? But maybe Carrie knows.
So Hedi, I'd like to ask you a question based on that. So I think this comes back to consent agreements. So when a project is first started, what are the consent agreements? And from ELIXIR, we do a lot of work to try and harmonize consent agreements across Europe where we can. So if we look at the consent agreements from Estonia and the Estonian Biobank, is there more than one type? And do they state, yes, I'm happy to give all of my data to international access to all different types? And what is the granularity of those consent forms? Because ultimately, I think that's where the question from Raul really sits. It's in the consent of the data. So
I don't know the very details of the consent form, but I think that it is that they give the right to the genome bank to do whatever they want, and but I don't know if it's specified, like, and also the other research projects, because Estonian Genome Bank is working together with, as far as I know, with some companies from states as well. But of course, the question is, can the genome data leave the country and things like that? So that's a tricky one. But at the same time, I think the people, themselves, they could export their genome if they would really like to. But it's a good question. I think we should maybe discuss that offline, because, and I would need to dig into that a bit to know the answer really.
Yeah, it was a great question, Raul. Thank you for that. Questions from anyone else? Attendees? Raul, did you want to comment? Sorry.
Yeah, I think it's an excellent opportunity for knowing ourselves better, and I'm hoping to have a good advance into the future together. Thank you.
Thank you very much. Gary, we have a question about how easy it is to implement a beacon. So say if a Romanian research group wanted to light a beacon, will they get technical support?
So capacity building is a key aspect of a sustainable infrastructure, and ELIXIR Beacon is a key product of ELIXIR. So we want to have as many beacons lit as possible, and we want to make it as easy as possible. So there's technical support at various levels. So there's webinars that are online where you can see how people have implemented them in the past, and we have some from CRG in Barcelona that show you how to use the reference implementation against a VCF file, for instance. If you don't have a VCF file as your backend variant warehouse, then there are online documentation. There is online documentation within the GitHub repo. So that's technical support, but as a documentation level. And then we also have bi-monthly meetings of the ELIXIR Beacon project that are focused on technical use cases, where there's an opportunity to come and talk directly to the people that run the specification and can help you there. So yes, technical level support is available at whichever level you need it. Right at the beginning, if you wanted to have a webinar, documentation, if you're technically au fait with the programming languages and could use that yourself, or come and speak to us. So I think all three options are there. I would encourage anyone that wants to light a beacon to really take one of them. And if that doesn't resolve the issue, contact me and I will do everything I can to help you light a beacon, because for this federated infrastructure to work, we need the beacons to be lit.
Excellent. Thank you, Gary. I think that's a really nice message to finish on. So it's 12:20 in Romania now, and I think we're going to close the webinar so that people can go and get some lunch. I'd like to thank all of the panelists for your preparation, your presentations, and being able to answer these questions as well. So thank you. I'd also like to thank all of the attendees for joining. We hope that you found it useful. We're available afterwards if you've got follow-on questions. And I'd also like to really thank Bogdan and colleagues from the Romanian Society for Bioinformatics as well, because it's a joint event, and this event couldn't have happened without them. So for those of you who may also be interested in plant sciences, I'm not quite sure the overlap of human data to plant sciences, but we do have one final webinar in the series. So perhaps we'll see some of you next week for that. So thanks a lot for your time, and hope you found that useful. Thank you.